Okay, so I'm going to talk about, I wanted to point out some of the features, some of the structures on a few of the models. So this is a neuron model from the lab, and this is a multipolar neuron. I know that because here's the cell body with our nucleus. Here are lots of processes coming off of it, so it's multipolar. Now, this is the cell body or the soma where it is funneling off and forming this elongated process. This is the axon. So this is the axon hillock, and this is the axon. Now, just pretend that this is attached up here, and this is a continuation of the axon. So it's just broken up into two pieces, but just take this and connect it on top over here. Okay, so here's our cell body, again with our nucleus. These processes that are coming off of the cell body, these are called dendrites. And the dendrites in the cell body are the receiving end of the neuron. Receiving meaning they are the ones that will be able to respond to a stimulus. And you see these little orange structures coming down and synapsing with the dendrites and the cell body. These are actually axon terminals of what would be presynaptic neurons. Presynaptic meaning before the synapse. And so this would be our postsynaptic neuron after the synapse. So these represent synaptic terminals, or axon terminals forming synapses from the presynaptic neuron and forming synapses with our postsynaptic neuron on the dendrites and the cell body, which are the receiving end, receives the stimulus. Then the cell body decides what it means and how to respond. And if we meet a particular threshold, then we have an action potential which will get sent out through the axon. So the axon is the outgoing part of the neuron. So the outgoing message will travel through the axon in the form of an action potential down to its synaptic terminal and will synapse with either another neuron or an effector like a muscle or gland. Okay? All these little broken up pieces, these little blue pieces inside of the cell body, these are called Nissl bodies. These are portions of rough endoplasmic reticulum. Lots and lots of protein synthesis happens in these neurons because all of the protein channels that have to be embedded in the axon for action potentials to take place. So here's our cell body, axon hillock. This is the axon, which is the outgoing. This is where the outgoing message travels down. Okay. You will see, so these cells, so here's one cell, here's another cell, here would be a third cell. The cells that are surrounding the axon, these are called Schwann cells. I know these are Schwann cells because it's one cell wrapped around one individual axon. Oligodendrocytes, remember, the cell body with its nucleus would be in between multiple neurons and then have processes that wrap around multiple axons. So these are Schwann cells. These are neuroglial cells of the peripheral nervous system, and they form this myelin sheath. So if you look right inside, so this has been cut away, like a sagittal section, and then you can see these lots and lots of layers. This is the myelin sheath, which is produced, so these multiple layers is specifically the myelin sheath, which is produced by these Schwann cells. And you can see that all this myelin causes the nucleus and other organelles to be pushed to the periphery of this Schwann cell. This myelin sheath, composed of myelin, which is about 80% lipid, 20% protein, it helps insulate the axon so that an action potential cannot travel where there is this myelin sheath. So the action potential actually has to hop from node to node. So this space right here, this is a node of Ranvier, this would be a node of Ranvier, and these nodes are where the action potential can actually take place, and this jumping is called saltatory conduction, and that speeds up the rate of the action potential. So myelinated neurons will be faster at communicating than unmyelinated neurons. If you ever heard the term white matter is referring to tissue in the CNS that is primarily made up of myelinated axons. Myelinated axons because this myelin sheath primarily lipid, and lipid looks white. whereas gray matter is the tissue in the CNS that is primarily dendrites, cell bodies, and unmyelinated axons. So dendrites and cell bodies are never myelinated. Let's see, so we talked about the nodes of Ranvier. This gray connective tissue that's covering an individual neuron, right? So this is an axon of an individual neuron. This would be endoneurium. Okay, so endoneurium because it's the connective tissue covering of a single axon. Here, so this is a spinal cord, spinal cord model. Out here, where these two roots merge, these are called spinal nerves, and the spinal nerve is made up of lots and lots of axons, of lots and lots of neurons, and so this nerve is surrounded by connective tissue that would be called the epineurium. So the epineurium is the connective tissue that covers an entire nerve. Endoneurium covers an individual axon of an individual neuron. Now, none of our models show fascicles surrounded by perineurium. That was on that previous recording that you had to listen to. But you can see epineurium and endoneurium. Okay, so spinal cord, white matter on, so this is the spinal cord proper, white matter on the outside, gray matter on the inside. White matter, you can call them funiculi, funiculus or singular, or columns. To me, columns, easier to remember, and it's actually more descriptive as far as what the function is. And then the inside little butterfly is the gray matter, okay? In the brain, it's reversed. Gray matter is on the outside, white matter is on the inside. So the first thing you have to be able to orient yourself with the spinal cord and know what is dorsal / posterior or anterior / ventral. And from there, naming all the structures is a lot easier. So, do you see these bulges? So in these two roots, this root has a bulge, but these do not, okay? Where you have this bulge is called the dorsal root ganglion. So a ganglion is a cluster of cell bodies outside of the central nervous system. So this being in the peripheral nervous system, these are nerves, all right? If you ever hear the term nucleus or nuclei, not like a nucleus in a cell, but a nucleus in terms of the CNS, that's a cluster of cell bodies within the central nervous system, okay? So this dorsal root ganglion is where the cell bodies of sensory neurons are located. And so if this is the dorsal root ganglion, guess what? This is the dorsal root ganglion. So this is the dorsal side, right? If this is the dorsal side, that means this is the ventral root, this is the ventral side, okay? You can say dorsal or posterior means the same thing for us. You can say ventral or anterior means the same thing for us. So I always identify this dorsal root ganglion first, and then I know this is the dorsal side and this is the ventral side. Dorsal root contains axons of sensory neurons. The ventral root contains axons of motor neurons. And where the dorsal root and the ventral root merge, this is called a spinal nerve. These are mixed nerves that contain both sensory and motor. Sensory information would be coming in from the peripheral nervous system, sensory neurons in through the dorsal root, and are going to synapse with motor neurons in this gray matter, and then the axons of motor neurons would exit through this ventral root, merging with the spinal nerve, and will eventually make their way to muscles or glands, okay? So sensory information in through the dorsal, motor information out through the ventral. This structure right here, which is a very tight groove, you can see very little space. This would be the posterior median sulcus. Sulcus is a tight groove, posterior side down the middle. Right here, where it's a deeper groove, where you can see space, this is the anterior median fissure. Anterior because it's on the anterior side, median down the middle, fissure is a deep groove. Okay? Now, the columns, the white matter, also called funiculi, the columns It's white matter, so it's primarily myelinated axons. These are myelinated axonal tracts that are going to and from the brain, so between the peripheral nervous system and the brain. So think of columns on a house. Columns go up and down. Well, columns in the spinal cord are axons that are going up and down, traveling between the brain and the peripheral nervous system. So the spinal cord serves as a conduit for axons going to and from the brain, right? That's one function of the spinal cord, is to serve as a conduit for information going to and from the brain. That's what these columns do. So these two structures right here, these would be the dorsal columns because they're on the dorsal side. These two structures right here would be the ventral columns because they are on the ventral side. And then this and this, these would be lateral columns because they are on the lateral side of the spinal cord. Now, the little gray matter butterfly, these are called horns, okay? This is where synapses take place within the spinal cord. Also, basic reflex processing happens here. When I talk about basic reflexes in just a second, the reflex arc, this is a way for your body to help minimize damage to peripheral tissues without having to go to the brain first. So when you put your hand on a hot stove, you yank it away really quickly, and then you realize that that hurt and that it was hot. That's because first, your spinal cord carried out that very basic reflex, that jerk reflex, and then the information traveled through these columns to your brain for processing so that you understand that was hot, that hurt, I don't want to do that again, right? But that reflex, when we want something to happen really quickly without our brain processing, without having to take the time of that information going all the way to the brain and then back, okay? So basic reflexes, as well as a conduit for information to and from the brain, those are the functions of the spinal cord. So here and here, these are the dorsal horns because they're on the dorsal side. Here and here, these are the ventral horns because they are on the ventral side. And here, these are the lateral horns because they are on the lateral side. Okay? This stretch of gray matter that connects the two butterfly wings together, this is called the gray commissure. This hole in the center of the gray commissure, this is the central canal. This is where CSF, or cerebral spinal fluid, flows. So here's our basic reflex arc. Remember, there are 5. You need to know the 5 steps. Step 1, you have to have some sort of stimulus. Okay, so this is that, damage to your peripheral tissue. So there is a stimulus, right? Then that stimulus is picked up by a sensory neuron. That sensory neuron is going to bring in that sensory information through the spinal nerves, then through that dorsal root, right? And look, this has a one process that splits off of the cell body. That cell body is located in the dorsal root ganglion. So remember, a ganglion is a cluster of cell bodies in the peripheral nervous system. This is that unipolar neuron from the previous PowerPoint presentation. Okay, this is a unipolar neuron cell body with one process. And remember, I said unipolar neurons are primarily sensory. And so look, bringing in sensory information. So step 1 is a stimulus. Step 2 is that information has to travel along sensory neurons into the gray matter, so through the dorsal root into the gray matter of the spinal cord. Step 3 is information processing within the CNS, so meaning this sensory neuron is going to synapse with the appropriate motor interneurons and motor neurons within the CNS. So there's information processing, very basic information processing, but it's still information processing. So we have these synapses taking place in the gray matter of the spinal cord. And then step 4, we have to have our motor neurons activated. So here's our motor neuron, and this is a multipolar neuron. Remember me saying that multipolar neurons are primarily motor? Here's our cell body with multiple processes coming off and one long single axon. Okay, so synapses with a motor neuron, motor information exits the spinal cord through that ventral root out through the spinal nerves, and it's going to synapse with a muscle, okay, with a skeletal muscle. And then that muscle is called an effector, and we have a response by that effector, which is step 5. So we have to have a stimulus. We have to have receptors on the sensory neuron to receive that stimulus, 1. Sensory information brought in through the dorsal root into this gray matter of the spinal cord through a sensory neuron, step 2. Step 3, information processing, so synapsing in the gray matter, step 3. Step 4, information goes out through the ventral root along motor neurons. Those motor neurons synapse with a muscle. And step 5 is response by the effector, okay, which in this case is a skeletal muscle. So this model from lab is showing just that, right? So here, these are free nerve endings that are located up in the epidermis. So here's our stratified squamous epithelium of the epidermis. Here we have free nerve endings going up into the epidermis, little Meissner's corpuscles. So it receives a stimulus, right? So this would be where our receptors are located. It receives a stimulus. That information travels along this sensory neuron through the spinal nerve. It's going to branch and go through the dorsal root. This would be our dorsal root ganglion where our cell bodies are clustered. This is a unipolar neuron. Into the gray matter of the spinal cord where we have information processing happen. Synapses with a motor neuron, which is then going to send motor impulses out through the ventral root, through the spinal nerve, and then synapse with a muscle, and then we have the response by that effector, which is the muscle, okay? This example is showing you a polysynaptic ipsilateral reflex arc. So this is an entire reflex arc with its 5 components. Polysynaptic because we have 2 synapses. So in order to have a polysynaptic reflex arc, we have to have an interneuron, right? So this interneuron makes it have more than 1 synapse. So this is a polysynaptic, and then ipsilateral because it is on the same side, right? So ipsilateral on the same side. Polysynaptic because there's more than 1 synapse. This is showing you an example, that knee-jerk reflex, when the doctor hits your patellar ligament with a little reflex hammer and you have a knee-jerk reaction. This is an example of a monosynaptic ipsilateral reflex arc, right? So sensory neuron coming in, synapses with 1 neuron, which is a motor neuron goes out and synapses with a muscle. So it's on the same side, so it's ipsilateral. And it's monosynaptic, mono meaning 1, only 1 synapse. Okay? This is showing you an example of a polysynaptic ipsilateral, which is a withdrawal reflex. So polysynaptic because look, we have this interneuron, we have more than 1 synapse. Ipsilateral because it's on the same side. Okay, so this shows a foot stepping on a tack, receives that stimulus, sensory information comes in through that spinal nerve, dorsal root, synapses with an interneuron, which synapses with a motor neuron, and then comes out through the ventral root, synapses with the muscle, which is the effector, so that you can withdraw your foot from that tack. Okay. A lot of times on the test, I don't ask you to diagram this really, more so as if I put this picture, an unlabeled picture, and I ask, what type of reflex arc is this? You should be able to identify that it's polysynaptic because there's multiple synapses, and that it's ipsilateral because it's on the same side. You should also be able to label what is the stimulus, what is the sensory neuron, where does information processing happen? What is the motor neuron? What is the effector? So you should be able to label the 5 components of a reflex arc, and you should be able to identify whether it's monosynaptic ipsilateral, polysynaptic ipsilateral, or polysynaptic contralateral. Okay, so contralateral meaning it goes to opposite sides. Okay. it goes in from one side and out through the other side, so opposite sides. All contralateral reflex arcs are going to need to be polysynaptic and have that additional interneuron on the inside. So this is showing you that reflexes can be a little more complicated than what we previously saw with the monosynaptic ipsilateral and polysynaptic ipsilateral. Is if you were standing upright on both legs, you step on a tack with one leg, right? That stimulus is received, sensory information comes in through the sensory neuron, through that dorsal root, synapses with interneuron, which then synapses with multiple motor neurons, and those motor neurons are going to exit through the ventral root and then eventually synapse with their effectors, which are muscles. But we have to have synapses with muscles on both sides because we're going to have a certain set of muscle, like our extensors, we'll have to relax on this leg that's being pulled up, and our flexors are working, right? So our flexors, our knee flexors are activated, but these extensors have to relax. Whereas on this leg, which has to, withstand the weight of our body when we pick up this foot, it's opposite happens. We have to have extensors contracting and flexors relaxing. Am I going to ask all of that detail? No, but you should be able to, if I show you this diagram, be able to tell me that it's a polysynaptic contralateral reflex arc, because look, sensory information coming in through one side, and we have motor information exiting through the other, okay?